So what is genetic or gene engineering? Nope, not those kinds of genes, and not that kind of engineering. Okay, so genetic engineering is changing the characteristics of an organism by altering its DNA. Animals, plants, microbes, and people, every living thing that contains DNA. Portions of that DNA make up genes. Genes code for proteins. Proteins play a huge role in giving an organism a physical characteristic, aka a trait. For example, let's consider your own eye color. Human eye color is a complex trait that's determined by more than one gene. Genes code for the proteins involved in producing that eye color pigment. But proteins coded by genes do a lot more than just determine your eye color. Proteins are involved in machinery of transport, in structure, in making and breaking things, in protecting the body, and so much more. So when you change the DNA of a gene with genetic engineering, you also change the associated protein, and that gives an organism a new trait. Genetic engineering might sound like some crazy science fiction mad scientist thing, but humans have been modifying the DNA of plants and animals for a very, very long time, before we even knew what DNA was. Early plant and animal breeders would choose individual organisms with desirable traits and use selective breeding to achieve the results they wanted: bigger and sweeter fruit, or dogs that were friendly and fluffy. These methods might sound like nothing but farming, but they are a form of genetic engineering nonetheless. As we've learned more about the science of genes, we've developed new tools that let us change DNA faster and more precisely. These tools let scientists add, delete, change, or turn on or off specific genes to achieve the more desirable characteristics in an organism. And since the genetic code is universal, we can even mix and match DNA from different species to create new combinations of DNA, or recombinant DNA. So you might be thinking, genetic engineering, never met it. But we use genetic engineering all the time, especially to treat and cure diseases. One of the most famous examples of this is how we've genetically engineered bacteria to produce the human hormone insulin, so we can harvest it and use it to treat diabetes. That's right, all we have to do is change the DNA of bacteria to make them do our bidding. What other tools do scientists use to change DNA? How do we get that DNA into bacteria and other organisms? And what useful things can cells do once they get that DNA? Here's a hint: lots of things. Next stop, what's in those scientists' toolbox?